Here we go. Here's your YouTube lecture for 9A2, which, as I said, is the meat and potatoes of this chapter. If you want to understand this chapter, this is the PowerPoint you want to focus on. Here we focused on our nested dolls, and we have nested dolls in terms of wrappings or connective tissue, and we have nested dolls in terms of muscular tissue itself. Muscular tissue. From deep to superficial. Now maybe we should switch this and do this from superficial to deep, going out to in. The outermost covering you have is called epimysium. Epi means on top of. The epimysium covers the entire muscle and protects it and ties it nicely together, again like a sausage wrapping. Then within the muscle you have a perimysium cover. Peri means around, which covers the bundles of cells, the cell bundles, which are called fascicles. Then you have the endomysium wrapping. Endomysium means inside, and that covers the individual cells or muscle fibers. Oh, actually, I guess I wanted to underline this ideally. This is a view of that, similar to your coloring sheet. Again, don't forget the purpose of the tendon here, to attach the muscle to the bone. Here's your epimysium, which covers the muscle. Then you have your perimysium, which wraps each fascicle. And then endomysium, which covers each muscle fiber. As I said, we're going to dig deeper into the muscle fiber. I made a mistake with the first class that I will rectify here. For the other classes I'll be reviewing. So in terms of muscular tissue, you have the entire muscle itself, like the biceps, the triceps, your lats. The muscle is made up of bundles of cells called fascicles. That bundle is made up of individual muscle cells or fibers. Those fibers, it's a cell, so within the cell it contains your normal cell parts, but in a muscle cell, because of its specialization, we're going to stuff that cell with these special organelles or fibrils. So now we're down into the organelle level. Fibrils are made up of proteins which help with muscle contractions. These proteins are referred to as filaments. And those filaments are of two types. There's a thick filament and a thin filament. So there's your hierarchy that you are responsible for muscular tissue. This slide is relatively generic. So a filament is a protein, a fibril is an organelle, a fiber is a cell. So if you keep going up above that, eventually when you get to a muscle is considered an organ. So if you recall from chapter 1, if I ask for an example of an organ in the muscular system, the biceps is an organ. If it's an organ, it's got to be made up of multiple kinds of tissues, not just muscular tissue. So muscle, in addition to muscular tissue, also has a nerve, has blood supply. That's established here. And the rest is kind of generic. The one thing here, I guess, you want to be aware of the difference between a nerve and a nerve ending. So, you have one main nerve, but then that nerve would branch into many nerve endings, which would eventually, each nerve ending would reach each cell to give it the signal to contract. This was discussed when we discussed joints. The insertion moves toward the origin. Here's our tendon, which is an indirect connection. The muscle becomes a tendon. The tendon attaches to the bone. This gives some protection to the muscle. The tendon is sturdier than a muscle and is not as likely to tear. There are some cases of direct attachment, again, but this is usually rare, as a tendon is going to allow that muscle to be protected. An aponeurosis you can think of as a very large tendon. These are present on the back, for example, where you have large sheet-like muscles, like the latissimus dorsi. We had discussed when we saw the ACL repair surgery how for the replacement for the ligament, sometimes you'll take a piece of a tendon off of the individual and then slice a piece off and then use it for the ACL. A good place to find that would be in an aponeurosis, which would have an excess of tissue. Again, I mentioned this figure, table 9.1 in your book. I would really open up that textbook, dust it off, and look at it. Look at this figure in it. Here's our 3 connective tissue wrappings. Here's our ham, or our muscle. Inside that muscle is our fascicles. If we zoom in on one fascicle here, we see that that's made up of a large collection of fibers. And if we zoom in on one fiber, we see that that's made up of a large collection of fibrils. Now, you're not responsible for the myo part. Save you 3 letters. And we're going to even dig into the fibril on the next the continuation of this diagram. So here we see that the fibril, I'm going to jump the sarcomere for a second, although this guy is very important. We'll come back to him. And the fibril is made up of, again, I'm going to jump this, of filaments. And here are your 2 types of filaments, thick and thin, which the specific protein that makes up the thick filament is named myosin, and the specific protein excuse me, that makes up the thin filament is actin. As far as the sarcomere, this is the functional, the contractile or functional unit of a muscle. So when your muscle contracts, at its most basic level, what's contracting is a million sarcomeres. So a sarcomere is more of a functional reference, which structurally consists of a combination of thick and thin filaments. Alright, so let's go through some of that here. Fibrils, again, I would consider these organelles within the fiber or within the cell. And again, a muscle, especially if you're ripped, would have a ton of fibrils. Those 2 filaments, thick and thin, they alternate in how they're, or I guess staggered within the muscle. So you get this alternating series of dark and light bands, which make up the striations you see in skeletal muscle. The darker part of the muscle is called, the darker stripe on the muscle is called the A band. The lighter stripe on the muscle is called the I band. The A band is dominated by the thick filaments. So I like to think of A as in fat, and the I band has only the thin filaments. So I like to think of I as in thin. Here's a zoom in of here's your 2 bands alternating, dark light, dark light. So this would be one fiber or one cell, and packed with fibrils. So each of these would be a fibril, right? All these in here, fibril, fibril, fibril. The one other thing I'll say here, which I believe, again, comes up on one of your tests at some point, SCO, quarterly, something or other. you'll see many mitochondria scattered within the cell here, and a skeletal muscle is packed with mitochondria because of the large energy requirement, the large amount of energy that a muscle cell uses when it contracts. All right, we get into the actual sarcomere here, so we'll break down that sarcomere and those A bands, those I bands a little bit more here with a couple other letters to broaden our alphabet. Again, the sarcomere is a contractile unit. It consists of overlapping thick and thin filaments, which are bound, or the boundary of that sarcomere is what's called a Z-disc. And again, there's two types of filaments in this sarcomere, thick and thin. All right, here's our picture with our alphabet. The orange stripe here is the thick filament. The blue thin lines are the thin filament. Again, myosin is the thick filament. Actin is the thin filament. One sarcomere is made up of the region from one Z-disc to the next. So that's one sarcomere. Here's the next sarcomere here, and here's a sarcomere here. So in essence, you would say there's 3 sarcomeres indicated in this section of a fibril. The A band, again, is dominated by the thick filament, but you'll see there is a region of overlap, and that overlap is important, where the thin filament partially overlaps the thick filament. And then in the I band, there's only thin filament, and you can see that here. All right, 2 more letters mentioned here are the M line. The M line marks the center of the sarcomere. So there's one here, you got one here, and one here. Probably the most important letter here is the H zone. And again, I didn't get to it in the first class, I did in the last 2 classes. But on Moodle, you want to watch the 2 YouTube clips of the sarcomere contracting that are posted on the face page there of Moodle. The H zone is the one part of the thick band, the A band, where upon relaxation there's no overlap. So there's no overlap when the muscle is relaxed. That's important to have that region where there's no overlap, because when the muscle contracts, the thick filament grabs onto the thin filament and pivots it or pulls it in towards the center, towards that M line, and collapses that H zone. And if you imagine that happening all along the length of the muscle, in this direction and this direction, that's eventually what's going to lead to your entire muscle shortening or muscular contraction. We'll go through again some of that here. The thick filaments extend the entire length of the A band. The thin filaments, again, it's important to note here, extend part way, but only part way, again, upon relaxation into the A band. The Z disc is just an anchor, or marks the ends of a marker really marks the ends of the sarcomere. Another marker will be the M line, which marks the center of the sarcomere. So Z disc marks the end of the sarcomere, and the M line marks the center. That's all you have to know about those. And here is what I discussed with the H zone. And again, that's when the muscle is relaxed. Alright, so all that is shown here. So I'm going to jump this. The thick filament is composed of a protein called myosin. It talks about this, and you'll see in the picture on the next page, there's 2 tails and 2 heads, and they're interwoven, but to me, you can just kind of functionally think of it like a head or a tail and a head. This would be the head. And myosin looks somewhat, very much like a hockey stick. And that head is the important part. This head can pivot. And as it pivots during muscular contraction, it pulls that thin filament in towards that center M line, collapsing the H zone, leading to a shortening of the muscle. So here's your hockey stick. And so that'd be one myosin. So a thick filament is actually, a large pile of hockey sticks thrown down. The thin filament is composed primarily, or chiefly here, of the protein called actin. Its other components, I'm not going to hold you responsible for their names at this moment. The point here is there are active sites, so on the actin, which allow the myosin heads to attach to it. That's very important. So here's our actin. So we have a hockey stick, and to me the actin is somewhat like an olive. So if you look closely at the olive, you'll notice, if we just look at this one right here, and again, for the moment I'm going to ignore the names of these other proteins, but what the purpose that they serve, which I do want you to be aware of, is that they block the active site. So in this current state, the muscle, I can tell you, is relaxed, because if you look at the spaghetti here, it's blocking the hole, the pit in the pitted area of the olive. So in this picture, the active site is blocked. And under this scenario, the muscle is relaxed. And in the next PowerPoint, what we're going to see is that in order to allow the head of the hockey stick to bind or attach to the hole in the olive and pivot and pull it in towards the center, contracting the muscle, we just need to remove the blocking agent. My voice was probably just cut off. I've been trying to be good about that. All I said was that's how a muscle contracts, when you remove the blocking agent. This is zoom in here. I think is worthwhile to spend some time on. So what you have here is a portion of an A band. Actually, this is a large portion of the A band. Let me get rid of this. This entire length here will be considered the A band, what's shown here. So that's the A band. In the center of the A band, from here to here, you have your H zone, and not shown here, but you would have M lines, here in the center. So again, in this picture, the muscle is relaxed. The myosin and the actin are unattached. What happens when the muscle contracts is, again, you're going to remove the blocking agent on actin, which will allow the myosin to link up with the actin, and as it does, it's going to change colors here. I'll change color in a second. This head will pivot just a little bit this way. So we'll go from this kind of orientation and pivot to this kind of orientation. Here's our tail of the hockey stick. And it will repeat that over and over again. And I've been trying not to use this word, but I'm going to, because it fits so well, in a ratcheting motion. So you can have fun with that. So as it repeats this pivoting or ratcheting, it brings this thin filament in towards the M line, and this thin filament in towards the M line. Again, collapsing the H zone, in essence making it disappear temporarily while the muscle is shortened. Good luck.